Hi, let's go. So this is the project, and the first thing you're going to need to do on the project is download the data. And so here we go. You're going to go to Blackboard, just like that, and you're going to go to your biology, and then there we go. Project Bioinformatics. That's the one you want. Here are the things that you need, and the example is a completed example. It's okay. The map section is not great. The other sections are pretty good. But it's at least something for you to go by. And the things you definitely need are the bacterial genome, and then you're going to need these programs down here, and I put links to them, but you can also just Google and find those right away. So step one, let's figure out where's your DNA. So we'll download this thing, open it up, and give it just a second. So you will have each been in a group, and your group will have been assigned one of the pieces of DNA, and so let's figure out which one that is. So if you were group 1, then boom, that's your DNA at the top, and so on. It goes down to the bottom. Now the one I'm going to play with is called the example. There we go. And so if you want to see it exactly the way I'm doing it, I would say grab this example and follow along. But what you're going to be doing is doing the same process that I'm doing, but with your sequence. So let's start by copying it all. And let's go to our first program. And the first thing we're going to need to run it through is called ORF Finder, Open Reading Frame Finder. So let's do that. ORF Finder. And there it is. All right. And so we're going to paste it in here. And then ORF Finder will find tiny ORFs and big ORFs. And ORF is an open reading frame, which means something that could encode a protein. It's got a start codon and a stop codon and something in the middle. Now, we don't want the teeny tiny ones because usually we can't say anything about them. And so a pretty good way to do this is to kick it up to, let's just kick it right up to 600. And you can do the same thing with yours. So we'll hit submit on that and wait just a second while it brings it up. And then boom, there we go. And so what we've got right here, this is what your map is going to look like eventually. It'll be something like this, but it'll have more information on it. So we can take a picture of this so that we can use it later. And so I'm on a Mac, and so it is Grab. And there's a different screen capture program that you would use on a PC, and you can Google to find which one is right for your computer, but you're taking a screenshot of part of it. And so oh, they've updated it. Grab is now screenshot. Well, fine. Okay. And so I'm going to make sure I get the scale in here because we want a scale model of our section of the genome. And I'm going to make sure I get all the genes in there. So there we go. And it's got that ugly green thing in there, but that's fine. We can ignore that later. And so, boom. Select that. Yes. Capture it. And we've got it somewhere. And there it is. Bam. Okay. And so we can save this and we can use it later. That'll be very helpful for the person doing the mapping section. Now for everybody else, what you can do now is you can go and you can just blast these things. So first you want to make sure that you're going to deal with all of these open reading frames, because anything that's bigger than 600 nucleotides, which all these are, because that's what we said in the last step to only show us, any of those are potentially going to be real genes. And so let's figure out if there are any that people have seen before. And we're going to select all open reading frames. We'll mark subset, and then it's all open reading frames right there. That's fine. So now we've got them all selected. And now now it says the marked set is all 13 of them. That's good. If yours only says marked set of 1 or something, that means you just selected 1. You got to go back and do exactly what I did. Mark the subset and select them all. And now let's blast all of these things. Now you need to make one little change on here. So if you use the Swiss protein database, I don't like the way it shows up. And I'm going to show you how it looks with the non-redundant database. So this is the one that's kept at the National Institutes for Health in either Atlanta or Bethesda, Maryland. I'm not sure where this server is, but who cares, really? And so we're just going to change the Swiss Prot database to the NR database, non-redundant protein sequences, and then we're going to hit blast, and it's going to go. And this may take a full minute, something like that, but it's usually going to be pretty quick. And this is giving us the result for all of the open reading frames that we found. So in my case, I had 13 open reading frames. There we go, marked set of 13. And so it is going to show me what gene is most similar to each of those 13 open reading frames. And they're numbered on here, which is very nice. And so you can use, like, if I wanted to just look at open reading frame 4, there it is, open reading frame 4 right there, open reading frame 1, 12, whatever. So it's got a number on it, and the little arrow in there, you see that? That tells you which direction the open reading frame goes. Now, DNA has 2 strands, and you can have genes going left or right. They can be on one strand or on the other strand, and that's just pretty common. What we've got here is kind of interesting, though. I've got genes that appear to be going in both directions and kind of sit on top of each other. So the same sequence is readable in both directions. Chances are only one of these two is going to be the correct answer, if you like. One of these two will have lots of matches from other bacteria, and the other one will probably have no matches. If I'm guessing right here, I'm guessing all the ones going in this direction are going to be correct for my section. But yours could be going in either direction. You can't really judge it by direction. The only thing I would say is that in a typical bacterium, there is very little and usually almost no wasted space. So you would expect right where one open reading frame ends, see that open reading frame 2, you would have another beginning almost immediately. So 6 picks up just barely after 2 starts. And then look, 7 picks up right after 6, which is why, since they're being really efficient, I think that's probably the case. And then look, 3 picks up just after that, etc. On and on. Okay, let's check on our blast, and it's going okay. Now, blast may time out. It depends on what network you're on and what time of day you're looking, but it doesn't lose your search. And so if it comes up with some kind of blank screen, like it can't find the internet anymore, like here, we can turn off Wi-Fi for a second, and then it'll say, oh no, no internet, it's broken, terrible. That's fine. Your search is still there, and as soon as we get back on the web, I will show you how to figure it out. Okay, so blast. And if you hit recent results, and this also shows up at the top of that page that's updating, recent results keeps a record of all the things you've run in the last 24 hours approximately. And if you want to see it, so it says it'll keep track of whether they're done or still in progress. And if they fail, just run it again. Usually that's just a one-off thing. Just keep trying. The request ID right here, that's the one that takes you to the results. And so it's going to need a second to format those. Here's the other way to get to recent results right up there. And as soon as it's done, it's still calling up for how best to format this thing. You know what? Let's open up another tab. And let's pull that up. Well, maybe it's still searching, although it says it's done, and usually that means it is actually done. So, I don't know. We may need a minute for this to come in, but that'll be the next section. I'll show you how to deal with this thing. So for right now, we've got our little map, which is great, and we've started the process of working on the other sections. So, oh, there we go. Now we've got the sequence up, or rather the results up here, and this is great. So I'll make the next section where we show you what to do with this, and another section showing you what to do with the map section up here. But for now, we are good, and so on to the next part.